This is a production of Cornell University.
Well, good afternoon, everyone. And we're going to get started this afternoon, even though I know that there will be people coming on in. There's so many incredible, wonderful, and competing events that are happening over this wonderful reunion weekend that I know people will be coming in throughout our presentation this afternoon. So that's great. I'm Kathryn Boor. I am the Ronald P. Lynch Dean of the College of Agriculture and Life Sciences here at Cornell University, and welcome home to those of you who have traveled to be with us this reunion weekend. We're very grateful that you're here, and we hope that you have a weekend that you will remember long into the future. And I especially thank you for joining us here today for our panel presentation that is entitled Genomics and the Future of Agriculture. And I'd like to start this afternoon by giving you a little bit of background on the Liberty Hyde Bailey Lecture Series. And that is the Liberty Hyde Bailey Lecture Series that we are celebrating this afternoon. This is an annual feature of our reunion events here in the college. And this event features distinguished faculty who are working in key priority areas for the college. In previous years, topics have included, for example, why social media matters. And we also have taken a look at the debate on the role of GMOs in feeding our growing global populations. It is my hope today that this Liberty Hyde Bailey lecture will both deepen your understanding of genomics and help you see the future of agriculture. And further, we hope that this lecture today will help demonstrate the impact that the College of Agriculture and Life Sciences has had and will continue to have as we look toward addressing complex global issues. It's particularly fitting that the name of this lecture is Liberty Hyde Bailey. And Liberty Hyde Bailey was the very first dean of the College of Agriculture at Cornell University. And I think you're all aware that the iconic Bailey Hall right across the street here is named after our first dean. Now, Bailey had many roles throughout his lifetime. He was a botanist, a horticulturalist. He was a plant breeder, certainly a teacher, a very successful administrator. He was a lobbyist, a very, very prolific writer. He was an editor. He was a poet. He was a rural sociologist. He was a philosopher. He was a traveler, and certainly he was an environmentalist. It is not in any way an understatement to say that he single-handedly helped to create the science of horticulture. His tireless lobbying of the New York State Legislature led to the establishment in 1904 of the New York State College of Agriculture right here at Cornell. Bailey was an early visionary in terms of seeing agriculture as an academic discipline. And to him, the fundamental purpose of education was to serve the people. And he believed that the resolution of agricultural problems were every bit as important as resolving other cultural, ethical, and legal issues, and hence his bringing agriculture to the fore as an academic discipline. He led a remarkable life and a remarkable career, and his influence not only here at Cornell University continues to resonate to this day. Now, just like Liberty Hyde Bailey, today's panelists are pioneers in their fields. Not only are they conducting groundbreaking research, they're also helping to fulfill the College of Agriculture and Life Sciences mission of providing knowledge with public purpose to the people of the state of New York, to the nation, and around the world. Today's speakers, Susan McCouch, Greg Martin, and Jim Giovannoni, are not only pioneers in their field, they are all former alumni of Liberty Hyde Bailey Professor Emeritus Steve Tanksley's lab. Steve Tanksley is a giant in the field of plant breeding and genetics. And we are so honored that Steve is here today. And Steve, if you'd stand up, that would really be appreciated. So Steve Tanksley. Steve's work in molecular genetics revolutionized modern plant breeding by creating the first chromosomal maps of plants and by pioneering genetic techniques that are absolutely essential to development of new crops. His research paved the way for improvements in size, nutritional value, and disease resistance of major food crops. His work also helped to lay the foundation for the breakthrough technology that is known as marker-assisted selection, which is a critically important tool for plant breeding. Steve did his undergraduate work in agronomy at Colorado State University, and he earned his PhD from the University of California, Davis, where he worked on rudimentary genetic maps for plants. He came to Cornell in 1985 as an associate professor in what was then the Department of Plant Breeding and Biometry. Over the ensuing decade, he and his team developed tools that allowed for complete mapping of the tomato genome. And in 1997, he spearheaded the Cornell Genomics Initiative, which coordinated way more than 50 faculty members from more than 20 departments across the entire university to help develop synergies that were based on the university's strengths in the life sciences. Steve was elected to the National Academy of Sciences in 1995 and the Royal Society of London in 2009, and he was appointed the Einstein Professor of the Chinese Academy of Sciences in 2006. Steve retired from Cornell in 2010 to work at Nature Source Genetics, which is an Ithaca bioinformatics company that he founded in 2006. In addition to all of the other achievements that I've already mentioned, Steve won the Alexander von Humboldt Foundation Award, the Martin Gibbs Medal of the American Society of Plant Physiology, Israel's Wolf Foundation Prize in Agriculture, the Rank Prize, and the Kumho Award in Plant Molecular Biology and Technology. And this year, he was awarded the Japan Prize, which honors scientists, and this is in quotes, whose original and outstanding achievements are not only scientifically impressive, but have also served to promote peace and prosperity for all mankind. Steve was one of two honorees that were honored this year. Steve not only had the vision to transform the whole field of plant breeding, his legacy also includes mentoring the next generation of groundbreaking plant scientists here at Cornell and building the university into a world leader in genomics. And here's something that you may not know. Right now, by U.S. News and World Report, Cornell University is ranked #1 in the world in plant sciences, and to a very large extent, that is the legacy of Steve Tanksley and his team. So that's an incredible accomplishment for Cornell University, and thank you for that. So now, building on Steve Tanksley's legacy here at Cornell, I'd now like to introduce today's panelists, and they'll come up one by one to give their presentations, and then when they're finished with the presentations, we'll all be seated here at the front. But for now, I'll talk a little bit about each panelist, and then I'll invite Susan up to give the first presentation. So I'll start with Susan. And Professor Susan McCouch is one of the world's foremost experts on rice and rice breeding. She received her BA from Smith College, her MS from the University of Massachusetts, and her PhD from Cornell in 1990, during which time she worked in Steve's lab. She spent 5 years with the International Rice Research Institute in the Philippines before joining our faculty here at Cornell in 1995. Susan's research into the genomic variations of different varieties of rice has led to the development of new rice strains that are more pest and drought resistant and that provide higher yields for farmers. And very importantly, Susan's work has created a more stable global supply of this essential food source. Our next speaker will be Greg Martin, and Greg is a professor of plant pathology and plant microbiology, and he is an expert on tomato disease resistance. Since 2005, he has been the Boyce Schulze Downey Professor at the Boyce Thompson Institute for Plant Science here at Cornell. Greg received his BS, his MS, and PhD from Michigan State University, and he was a National Science Foundation postdoctoral fellow at Cornell from 1989 to 1992. And it was as postdoc that Greg worked in Steve Tanksley's laboratory. Greg's research focuses on the mechanisms that bacteria use to infect plants, and in turn, the mechanisms that plants have evolved to interfere with the bacterial pathogen. Our third speaker this afternoon is Jim Giovannoni, and he is an adjunct professor of plant biology in the School of Integrative Plant Science. He's a research scientist for the U.S. Department of Agriculture Agricultural Research Service, and he's a professor at the Boyce Thompson Institute. Jim earned his PhD in molecular and physiological plant biology at the University of California, Berkeley in 1990, and that was followed by 2 years as postdoctoral researcher in Steve's lab. So you see how this all connects. Jim was recently elected to the National Academy of Sciences, and let's give Jim a round of applause for that incredible accomplishment. Jim's research focus is on the molecular and genetic analysis of fruit ripening and the consequences of those effects on ripening on nutritional quality. So now I'm going to turn the podium over to Susan McCouch, and Susan, if you would join me up here, please.
So it's a pleasure to be here, and this is, of course, honoring Steve. It's a rare opportunity that we get actually to tell somebody who has influenced our lives so deeply how much we appreciate what they pass on. And I think we all know the importance of a professor, so it also resonates back to each of us as professors hoping to influence another generation of students. So, this is the Steve Tanksley I knew in 1986 when I joined his lab. We were in 418 Bradfield Hall in the Department of Plant Breeding and Biometry. Steve arrived at Cornell with big visions of what molecular genetics could do for plant breeding, for agriculture, and for global food security. And in 1986, the world faced many of the same grand global challenges that we face today, although I think the understanding of what global climate change would represent had not yet become embedded in our minds. In any case, Steve was bringing new tools and a new vision to the Department of Plant Breeding and Biometry, and molecular markers would revolutionize the field of plant breeding and of molecular genetics. Particularly interesting to me was that it had immediate application in the opportunity to translate the discoveries that were being made in some of the basic sciences into important products in agriculture, medicine, and environmental science. And to me, this was music to my ears. I had a background in history and literature. I was coming from a liberal arts background. I had lived and worked extensively in the developing world. And during my first few weeks in the Tanksley lab, I quickly realized that I was going to have to learn an entirely new language. It was one of the most exciting periods of my life. I joined the team working to create the first molecular map of rice, and with funding from the Rockefeller Foundation's program on international rice biotechnology, I joined two colleagues, Zhengyuan Wang and Zhihong Yu, who were recently emerged from the Cultural Revolution in China. This was among the first wave of scientists to come out of China, and we were very excited to have them in the lab working on the development of the rice map. It consisted of 135 RFLP markers, and it was published in Theoretical and Applied Genetics in 1988. When I finished my PhD in Steve's lab in 1990, I took a position as a rice breeder at the International Rice Research Institute in the Philippines, and I worked there for 5 years. During that time, we established the first molecular marker lab, which was called the Genome Mapping Lab, and it was the foundation for the application of genomics in breeding at the time, which was all devoted to the concept of marker-assisted selection. The lab remains productive to this day, And during the time I was there, one of the more exciting aspects of being in the Philippines, other than the fact that I had the rice paddies right outside my window, was the fact that we had the gene bank right on campus. And part of the opportunity was to start to mine that gene bank for natural variation that we could use in breeding. And many of the genes that are actually present in ancient land races and wild species that are housed in the Erie Gene Bank have now been deployed through the use of this technology, marker-assisted selection, not GM technology, but actually through backcrossing, have been deployed to confer heightened levels of disease and insect resistance, drought tolerance, submergence tolerance, and many other traits. And those varieties are now planted on hundreds of thousands of hectares across Asia, Africa, and Latin America. Other students in the Tanksley lab built on the work that we had done in rice, using the rice genome as an anchor to start to map other related genomes that were larger and more complex. The first comparative map was the rice-maize map. And as you can see in this diagram here, we have a rice chromosome in the middle, and we have two corresponding comparative segments that were conserved in maize. And the interesting thing about this is that rice is a very small genome, and it was a fairly conservative and fairly stable genome. So it provided a very robust framework for this comparative mapping to assemble these much more complex genomes. And it demonstrated that these related cereals, they went on to develop maps of rice, wheat, and maize, and eventually many other comparative maps. But these three related cereals, which are the foundation of the world's food supply, were mosaics of their last common ancestor. And they all shared large segments of chromosomes that were still recognizable after 3 million years of evolution, despite being rearranged and reorganized in modern crop genomes. For those of you who are familiar with this, it's not surprising today, but at the time it was really a very profound strategic concept that started to inundate the field of molecular biology and molecular genetics. And these comparative maps had enormous implications for application in both agriculture and medicine. Because of its small size, the rice genome was also the first crop to be fully sequenced, and it provided a scaffold that allowed us to more easily assemble the more complex maize and wheat genomes. Now, maize is 6 times larger than rice. It's about the size of the human genome, and it was a slow and difficult process to assemble the genome sequence of maize, but even more difficult was wheat. Hexaploid wheat is 30 times the size of the maize genome, and it was only just recently completed in 2014. In any case, the comparative maps laid the foundation for this major accomplishment in the field of the plant sciences, and I might say also paved the way for my return from Erie to join the Cornell faculty in 1995, because if it had not been that rice was the foundation for the more important crops in the West, nobody would have thought of investing a faculty position in a person who would devote their life to rice. In any case, with so much attention focused on genotyping and sequencing of genomes, people sometimes seem to forget the reason for doing it and the creativity and effort that would be needed to translate that sequence into products that would ultimately impact the human lives around the world. Now, I returned to join the faculty in 1995, and by 1995, things were starting to get underway at Cornell. Many people were involved in aspects of genomics, but everyone did it in their own lab, buying their own machines, working independently, and often having to learn very difficult lessons, each one on their own. And there was no forum for sharing information and technology across the campus the way there is today. And Steve at that time had started to think about what the implications of the discoveries in the basic sciences meant for institutions and institutional organizations. And I'm going to quote to you some Actually, I took this off of an overhead slide, and I thought of coming in and putting up the overhead projector so that you could actually see I still have these slides in my vast repertoire of old papers. And these were some of the insights that Steve shared with many of us at that time. The DNA sequence will soon be known for most major life forms, not just for the major species, but for most major life forms. And biology will move from being an information-limited science to a science that is limited only by our ability to organize and analyze information. And today we live in the era of big data and digital information, and we all know what a burden it is to have an overload of information. Biological research will shift from a reductionist to a synthesis approach. In fact, it has dramatically shifted from dissecting complex genomes to predicting and synthesizing future outcomes. More basic discoveries will emerge from biology in the next 30 years than from any other single discipline. Well, I joined Steve's lab in 1986, and here we are 30 years later. I would say we've had a pretty good ride. And one of the most lasting contributions that Steve made to the Cornell research community was his unswerving support and leadership of the Cornell Genomics Initiative. Under this initiative, we would initiate a new campus-wide hiring strategy that allowed candidates for faculty positions to interview with prominent scientists in genomic-related fields all across campus. We would form a set of core facilities that provides the entire Cornell community and survives to this day with access to the latest in genomic technologies at a cost that was finally manageable for Cornell researchers due to the economy of scale that came from uniting the campus. And departmental and college boundaries would be lowered as faculty, staff, and students started to get to know each other through their shared research interests. And the kind of consolidation and interactions that I think Kathryn mentioned cut across many, many different colleges, many different departments, and has affected the lives of many people who were either at Cornell or who were recruited to Cornell under the Genomics Initiative. When somebody says to you, well, you know, what you did for your PhD that took you 4 years and was 135 markers on a map is going to be done in 10 minutes. A few years later, you kind of look back at your thesis and you think, wow, those were a lot of late nights in the lab, manually running gels and waiting for things and turning them around. And all I can say is that it was hard to imagine what somebody at that time could have foreseen when we talked about being in a data-rich environment. But indeed, I just want to end by saying that currently, the largest project in my portfolio is called the GOBI project, the Genomics and Open Source Breeding Informatics Initiative. This is a project funded by the Bill and Melinda Gates Foundation. There are 4 PIs, principal investigators, from plant breeding and genetics alone, 1 from BTI and 1 from computer science department. The objectives of this project are to program the software that is needed to facilitate the use of big data in breeding. And the goal is to assist breeders in the public sector working to accelerate the rate of genetic gain for staple food crops in the developing world. So you can see how this thread has actually permeated my entire career here. And I just want to close by saying that the GOBI project encompasses 3 different international crop improvement centers, CIMMYT in Mexico, ICRISAT in ICRISAT in India, and IRRI in the Philippines. There are 3 institutions right here in Ithaca, all of whom have PIs on the table, the USDA, the Cornell, and that's CALS, and BTI. And we are addressing simultaneously 5 of the world's most important crop species: rice, maize, wheat, sorghum, and chickpea. And this is, in fact, I think, a consolidation of the vision that Steve expressed so many years ago, just to show you that Cornell still remains, I think, a leader in the field. And I am very impressed, Kathryn, by your news today, that the plant sciences is ranking so highly. I must say I'm not surprised. but I think it's important for us all to recognize the excellence that is housed here at Cornell. And with that, I'll close by saying, I think I've done my part to contribute to the next generation as well. So thank you very much.
Well, thank you very much, Susan. And next we will hear from Greg Martin. So, Greg.
Well, I'd like to begin just simply by thanking Steve for his mentorship and for really his friendship over a long period of time now. So I was actually also a beneficiary of the Cornell Genomics Initiative because it provided me an opportunity to return to Cornell. I first joined Steve's lab, as Kathryn mentioned, in 1989, which is hard to imagine that was actually 27 years ago. And then, of course, was then able to come back in 1998, I guess is what it was. So it's interesting for me, reflecting back in that period of time, back in the late 1980s and the early 1990s, it turned out to encompass a really interesting intersection between two important developments in my field, in plant pathology. And that is, it was the first development of new experimental systems, genetic systems, that allowed the genetic characterization of how plants respond to pathogens. And then, of course, the second important development were the methods of using DNA markers that was spearheaded in Steve's lab. And those two different developments, again, intersected and allowed then the identifications of genes from plants that conferred disease resistance. And that ended up being the basis of my career. So what I want to do today is to give you an overview, a very kind of broad overview of what we've learned in the past 25 years about plant disease resistance. And then I'll touch a little bit about how we can use this knowledge to make better plants, to make plants more disease resistant. So if anyone is a home gardener, you'll know that plants succumb to all sorts of different diseases. They attack stems, fruits, leaves, and not shown on this slide, but also roots. They, of course, have a huge impact. They can have a huge impact on yield, but also on quality. And if you're a large farmer, that affects the marketability of the crop. If you like this kind of thing, and I do, they're also really interesting to study diseases because you can learn a lot about how the pathogens actually cause symptoms, how they're able to suppress defense responses in the plant. So aside from the fact that it's just a lot of interesting biology and it's kept my lab occupied for about 25 years, of course, diseases also have a major impact on plants. So it's kind of hard for me to see this up here, but 10% of food production, it's estimated, is lost due to diseases. It's difficult to control diseases. It costs a lot of money and, of course, involves the application of a lot of pesticides. There have been introduction of important diseases into the United States that have had a big impact on all of us. The American chestnut tree was lost to an exotic pathogen that came in. And of course, we're now currently dealing with citrus greening, which is shown here. And then the problem, and it's just like the problem with superbugs that are in the news recently, is that plant pathogens also evolve very quickly, and they develop resistance to different pesticides that are currently being used. So the good news is that plants actually have a very effective immune system. And in some ways, it's analogous to the human immune system. And it's important that some plants actually have natural disease resistance in the form of certain genes that allow them to resist infection. So this is just a simple picture that makes the point that, in this case, both of these plants were inoculated with the same bacterial pathogen. But you can see that one plant appears to be completely resistant to the disease. It really doesn't show any symptoms, whereas the other one has really succumbed to this disease. So this is an important point, is that plants naturally have these genes that confer resistance. So where do they come from? Many of them have actually come from wild relatives of tomato. And these were collected back in the late 1950s and early 1960s by Dr. Charles Rick, who was at the University of California, Davis, and was Steve's PhD advisor. So seeds of these different wild relatives were collected and are available now for us to analyze. And that's the sort of work that we've been doing over the last 10 or 15 years, is screening these to identify new disease resistance genes that could then be introduced into tomato varieties. So how do we do this? It's a little bit complicated. I'm not going to go into all the details, but the main point is that we're able to take advantage of both sort of classical genetics and classical plant pathology, as well as a lot of the more contemporary genomics technologies, in order to very precisely map a gene on a particular chromosome, get down to a very fine level, and be able to pick out the individual gene among the 35,000 or so genes that are in tomato, the individual gene that's important, that is, that's actually conferring this disease resistance. So as Susan alluded to already, this sort of technology has really expedited things. 25 years ago, when I cloned a resistance gene, when I was a postdoc in Steve's lab, it took 4 years, and many, many people working on it. We recently cloned a gene, and it took 4 months. So that's the sort of progress in the field. We're able to identify these genes much faster, and of course that will benefit plant breeding in the future. So what do these resistance genes do? I've just sort of described this in a very simple way. We now know that resistance genes produce proteins, and these proteins allow the plant to detect the pathogen. So pathogens produce certain proteins in their process of infecting the plant, in the process of causing disease. And these proteins that resistance genes make, we refer to as receptors. They can act either inside the cell or outside the cell. Once they detect a pathogen protein, it activates the immune response in the plant and leading to disease resistance. So this is really kind of the basic thing that we've learned in the past 25 years. But you can imagine with so many people studying it in lots of different labs, we actually know a little bit more than what I'm showing. And that's illustrated in this slide. And sort of, again, actually a rather simplistic view of what we actually do know about the immune system. We know a lot of different components that act both in recognizing the pathogen and in the plant response, how it actually resists infection downstream. And so each one of these different sort of items on this slide represents a different potential target that plant breeders can use to then introduce into new tomato varieties. And that's exactly what they're doing, using the sort of genomics technology that was pioneered in Steve's lab back in the late 1980s. Plant breeders, in this case tomato breeders, are using genomics technologies, in particular DNA markers, to be able to very rapidly breed more disease-resistant tomatoes. So with that, I'm just going to end with this sort of a simple summary slide to tell you that that diseases, of course, do have a huge impact, both in agricultural settings, but also to the impact, the economic impact on the farmer. There's been a lot of progress in understanding exactly how the plant immune system works, and that's due to many different labs, and we've been fortunate to have really an excellent level of federal funding, at least in the past 20 years or so, not as much currently, and that's allowed a lot of progress in this field. And then, as I mentioned, the genomics technologies allow both the detailed study of the immune system, but they also facilitate the breeding of more disease-resistant plants. So I'll end with that. Thank you very much.
Well, thank you very much, Greg. That was great. And now we'll hear from Jim Giovannoni. So, Jim.
Okay, well, thank you very much. First, I'd like to, again, thank Steve. You really changed my life, and in a very positive way. So some people have changed it other ways, but you had a great impact. And I'd also like to thank you and Susan and Greg, because I never get to talk to so many people, so it's nice to have a big crowd also. So I'm going to, I guess, tell you a little story about some of our work, some of the implications of our work in fruit development and fruit biology. But before that, to really just emphasize that it all started with coming here to Cornell for a postdoc and coming to Steve's lab. At the time, as Susan and Greg mentioned, the technology, marker technology, was being developed and applied to both the utility in genetic selection for breeding purposes, but also towards understanding and identifying genes. So it's being used both for practical and for basic biological purposes. And at the times, the first targets that Steve was approaching and that Greg implemented was the cloning of the first disease resistance gene from a plant. But before that gene had been cloned, I had met Steve and said, look, I'm interested in fruit and these funny ripening mutants. And Steve said, yeah, maybe there's something about those disease genes that's not going to work out. So why don't you come and work on the fruit genes just in case? Because the fact is, disease resistance is much more important. You never get a fruit if the plant is sick. So that's why I ended up here, and it was a very good thing. Okay. So thinking about fruit, we all know that in meeting our needs for food security, having enough to eat in our state, in our country, in the world, we often think about yield. But there's another part of that equation, and Greg alluded to it, and that's how much we can keep, how much we can actually get to the table. So shelf life, storability, these are all important. Seeds are designed through evolution to last, right? They'll overwinter, they'll make it to the next year. But the plant organs are not necessarily so designed. And so fruit and vegetable tissues of the plant that we consume, often have much more of a problem in terms of storability, in terms of decay. And the fruit in particular is an interesting organ because it's one that's designed through evolution to be eaten. So you think about the fact that most of what a plant is doing, as Greg just mentioned, is defending itself. And suddenly in this stage of development, an organ is going just the opposite, saying, come here and get me, consume me. And to do so as to release the seeds, because a fleshy fruit has really evolved as a means of releasing seeds for the next generation. But that process that makes the fruit attractive, the ripening process, also eventually leads to decay. And that's the other part of our equation in trying to meet our food security needs. How much can we produce, but also can we get it to the consumer? So this process that I've mentioned or referred to as ripening is really the sum of all those activities, the physiological changes, the biochemical changes that sum together to make the fruit attractive to some organism that will consume it and liberate the seeds. And again, if it doesn't come along, Plan B is become susceptible to things that will rot the fruit and let those seeds out. So that's always the challenge. How do we get high quality and something desirable to eat without getting into that later phase of what's referred to as over-ripening and eventually decay? So fruits do a lot of things, and they don't all do the same thing. They accumulate different pigments, they become different colors, some get soft, some don't get soft, but they have many of the similar properties. So they generally accumulate sugar, they generally produce volatile compounds, often compounds, and these are what give fruits their characteristic aromas. They're attractants, they smell nice to you, they're often derived from necessary nutrients, so they're also signals that there is quality there beyond just something that's fun to eat. It's actually going to be good for you. They, again, typically accumulate pigments, but different pathways and different fruits are activated. And nutritional content often changes in the fruit as well. And finally, as noted in the corner, eventually they rot. This is all part of the same developmental process, the same genetically controlled program. So all of this is going on as the fruit reaches that stage of maturity. And again, the challenge is, how do we get it to the consumer without losing too much of the crop to decay? And we put a lot of money into this, a lot of energy, a lot of time. We hold things in modified atmosphere storage. This is an apple shed in the Pacific Northwest where fruit are held in modified CO2 and oxygen levels and at low temperature, and that allows us to keep them for 9 months beyond harvest and get them to market virtually all year round. But in certain parts of the world, often the resources are not available, or the means for sustaining quality product is not necessarily there. It's too expensive, it's simply not available, the energy's not there. So we lose a lot, and we lose the most in the parts of the world where we need that food the most. So it's a challenge to address this problem, and it begins with us for really understanding the basic biology of the ripening of the fruit. And we do this in tomato, and again, because really in Steve's lab, so much was developed in terms of molecular resources, things like the genetic maps, eventually the DNA sequence, so that Steve really championed the sequencing effort in tomato. And a number of other attributes of those of you who grow tomatoes in your backyards know it's pretty easy. So I can grow tomatoes and without killing them off. The genetics is good. Because it's an important crop, breeders have collected a lot of the type of genetic diversity of wild species that Greg mentioned, and also just a lot of mutants, because they see interesting things, they put them into the seed storage bank. So there's a lot of genetic material that we can use to ask questions about plant biology, and particularly the fruit. So I mentioned the sequencing effort. I'm just going to highlight this for a second because, again, this was really Steve's vision. And he got together the group that started the sequencing effort. And it was, tomato is not a crop that is that important anywhere for anybody to be able to do it by themselves. So he organized an international consortium that tackled this problem. And this is, all the tomatoes represent the countries where there was actually funding, starting with a white paper that Steve organized, that everybody submitted to their respective governments, got money to bring about this project. And many other countries who didn't have funded grants also participated as well. The US was really the lead group, and Steve is biggest here, not because he has the biggest head, but because he had the biggest impact. But what I point out with this slide, these are all the groups in the US, and many of the people here are people that either worked with or were students. Here's Zach Littman, one of Steve's former students, Andy Patterson, another former student, Rod Wing, a former postdoc. So many of the people who were working on this project as faculty had come out of Steve's lab earlier on. And the sequence was important not just because tomato is important, but because it's closely related to a lot of other important fruit and vegetable crops. Potato, eggplant, pepper, you combine those tomato and you really have the most important fruit and vegetable crops in the world. And even coffee is not so distantly related. And Susan showed you a while ago the comparative maps between the cereals that were done with rice and maize, but Steve had also done this between tomato and coffee, demonstrating that these genomes are not that different and that you can use one genome to make inferences about the other. One of the real great impacts of Steve's vision and his leadership. So I'll spend a few more minutes now kind of coming back to some of the things I did and put it in the context of how I ended up here at Cornell also. I did my PhD at UC Berkeley in the late 1980s, and I was working on a gene in tomato called polygalacturonase. And that's referred to up here with the PG. Polygalacturonase becomes very highly expressed in the ripening fruit. It's the predominant protein in the cell walls of the fruit as they mature. And what it does is it breaks down the pectin in between the cells. So think of a brick wall, and if all the mortar started liquefying, that's what polygalacturonase does. It breaks that down. It was thought that that caused the fruit to soften. And so a company you may have heard of called Calgene said, let's make a genetically engineered tomato that won't soften. It'll last longer, we can leave it on the vine longer, it'll taste better because we don't have to harvest it too early just so that we can ship it across the country. And I assume probably many of you have heard of the Flavor Saver tomato and Calgene, and that was the first GM crop. So I was doing my PhD, and it was actually to test the function of that gene, and my PhD was that's not going to work. So what we did was we did the opposite experiment. We overexpressed it in a hard green fruit and said, does it get soft? The answer was no. The hard green fruit we used were a mutation called the RIN mutation, or ripening inhibitor, that actually arose spontaneously here on the Cornell campus in Henry Munger's greenhouse. So Henry Munger, former alum and also a faculty for nearly 40 years, passed away recently, was in the plant breeding department here. And this mutation arose spontaneously in his greenhouse. The fruit do not ripen at all. That let me test that hypothesis, but it also made me much more interested in, well, what's wrong with this fruit that it won't ripen? And that's what led me to Steve's lab, because he was pioneering the technology that allowed one to go from a phenotype to the gene that underlies that defect. So that's why I was here in the early 90s. I was actually in the same aisle as Greg Martin. We were bouncing into each other, not as much as we would now. I was much smaller then, but I still did once in a while. So the RIN mutation, what I just showed you, and you can see again here, is a homozygous mutant. Fruit never ripens, so that's a pretty serious defect. It actually has practical utility, a lot of it. It's used very widely in commercial tomatoes as a heterozygote, where you now get a long shelf life, slow ripening tomato that nobody likes, right? Tomatoes never taste as good as they do when I grow them in the backyard. This is one main reason why. But they do have long shelf life. You can ship them across the country easily. And it's really partly because we haven't focused too much on the quality. We primarily focus on making sure we can produce the crop and get it to market. But a lot of the breeders now are focusing back on the quality of the fruit. But again, our interest was this must be something really important. If you can alter this single gene and have such a big impact on the ripening of the fruit. And so in our lab and work, I started in Steve's lab and then completed a little bit later. We identified the underlying gene. It's a transcription factor, so it's a master regulator of other genes. And it basically, we know now, controls almost every gene that we know is involved in ripening or has a change of expression in ripening. This gene sits on its promoter and influences its expression. So it's a key regulator. There are many other interesting tomato fruit mutants, and implementing these same kinds of positional cloning technologies that Greg mentioned earlier, we've identified many of those genes as well. In fact, virtually every important or kind of major effect mutation in tomato has now been cloned either by our group or in collaboration with others. And Julia Vrebeloff, who did a lot of this work, is a former Cornell alum. She received her PhD here. And one of the interesting things we've done is once we had this gene, we asked, well, is this just tomato or is it other species? Well, we know now, working with a number of collaborators, generating knockouts in this gene through GMOs in banana and melon and strawberry, that this gene's very well conserved, and if you alter its expression, you alter the ripening of the fruit. So it's a very basic regulator of ripening, and it's a very well conserved through evolution regulator of ripening. So if you're interested in controlling shelf life, variation in a gene like this is probably one you should be looking at. As I mentioned, there are a lot of other interesting mutants. I'll just point out one here that we've identified the gene for. Another Cornell alum, Kong Wen, did his PhD on this. This is a gene called uniform ripening. So this is a wild type, non-mutant tomato. It has this kind of green shoulder on the top. Most tomatoes you'll find in the supermarket carry the uniform ripening mutation, so they're uniform in color. When they ripen, they'll be uniform red. The older varieties, and you know if you're interested in heirloom tomatoes, one marker of an heirloom is if they have the green shoulder. It's thought that that's not a nice appearance, so it's been bred out using the uniform mutation. What we know now is this is another transcription factor. That greenness up here is, of course, due to chlorophyll. If you remove that, you have less chlorophyll, you end up with less sugar, you have less chloroplasts, so you also get less carotenoids. You actually have probably a less nutritious fruit, but it looks nice. But it's an unintended consequence, and we understand this now. And we can probably do things about that because of that knowledge to bring back some of that quality. As Greg mentioned, we're also using genetic diversity now to look at, to identify additional genes for which we don't have mutations, but for which there is diversity in the wild germplasm. And this is just an example of a population that was developed largely using the marker technology that Steve had developed. And he's also developed some of these populations in his work as well. Here we're just looking, we're interested in the nutritional quality, and one of the main attributes of that in tomato is the carotenoids, lycopene, beta-carotene. That gives them their typical orange reddish colors, and I think you can see just looking over this slide, there's a lot of variation. If we extract those and measure them and just plot the different compounds across the different lines, you can see a lot of variation in different nutritional compounds. So we're using that now to map new loci and identify new genes that regulate this particular pathway and the outcomes. And again, this was another Cornell alum, Ryan McQuinn, who received his PhD here just last year, who did a lot of this work. So I thought I'd end, if I have a minute to end, with just one last slide about, who are our greatest faculty. And of course, we can replace this word here with Steve Tanksley, these two words. So he fits all of them, and we didn't coordinate, but you heard a lot of these words from Susan, from the dean, from Greg earlier. So I'm glad to see we're all thinking of the same things when we're thinking of Steve, an educator, an innovator, not just in science, but also in the university community and how science is done. Vision to see things. I think Susan showed that very well with her slide. When we were sitting here running gels, it was hard, I think, certainly for me to imagine where we would be now. And I think Steve saw that coming and helped bring us there. So that leadership as well. And the most important thing, I think, is still a human being in the end, a very compassionate person. And also sometimes he helps keep you out of jail. So I'll tell you a little story. It's not as bad as it sounds. But when I was a postdoc here, I was out in the field in Varna, where Varna is down 366. Steve had his tomato fields there, and it was a Saturday morning, and I was out with another postdoc, Rod Wing, and we were harvesting tomatoes from our crosses. So these wild species crosses, they make little tiny tomatoes. And along came a Cornell University police officer, wondering what we were doing out there on a Saturday morning picking tomatoes. He thought we were stealing them. He didn't seem to buy the fact that across directly 12 feet away from us was Martha Muchler's field for breeding tomatoes that had these big, beautiful tomatoes, and we had all these little tiny orange tomatoes. So he gave us a test. It's not the kind of test you see here, but it turned out was a recent Cornell grad. He gave us a plant physiology test in the field. We couldn't believe it, and the funniest thing was both Rod and I were, do you know that? Do you know? Started asking us about photosynthesis, about hormones. He had taken a hormone from Peter Davies. Very excited young police officer, and after that, we told Steve, of course we tried calling Steve. There was no cell phones in those days, so he was calling into the police station to call Steve in the lab, who was in the greenhouse. So we couldn't get rescued by Steve, but after that, we always carried a letter from Steve out in the field saying we actually worked from him and we were not tomato thieves. So that was quite an experience. So I will end there and just thank again all of you for coming today, and thank Steve for so much.
And now, if I could ask Susan and Greg to please join us all up on the stage. I have a series of questions that I'll be asking our panelists, and then I'll open up the conversation to everyone who's here today. But I cannot tell you how happy I am to finally learn the answer to the riddle. why those supermarket tomatoes never taste as good as the ones you grow yourself. So Rin, I've got that now. That's terrific. So anyway, let's start our conversation here among our group. And the first question that I'd like to ask our panel here is, what do you see on the horizon? What are the number one challenge or question I'd like to hear from each of you that you see on the horizon that will be important for plant scientists to research and address in the coming decade?
Just like that.
Just like that.
So one of the things that I think is really exciting is how to develop and deploy diversity, meaning genetic variation, in time and space in a way that will help us mitigate the effects of the variability of the climate. So if you can't predict something, you can't really work with a uniform agricultural system. You have to now reintroduce diversity. How are we going to do it? What's it going to look like, and how are we going to manage it so that it's actually both productive and appropriate for the challenges at hand?
Well, I think I tried to sort of illustrate this in my talk. In my field, I think the biggest challenge is how we're now going to start to take advantage, really, of what we've learned over the past 20 or 25 years about the immune system. We know a lot about it. We know about the individual genes, but we're just at the sort of inflection point of now starting to use those to improve disease resistance in the field. And that's going to be really important. I mentioned also superbugs in the news. That does happen with plant pathogens. And we need to be able to understand how to actually use multiple resistance genes in the same variety to be able to thwart the attack of these kind of superbugs that are always evolving out there.
I guess, one way I would answer that is what keeps me up at night, which is, what do we do with all the data? We generate, and this is, I speak for myself, we generate so much data that we maybe pick a few cherries from or analyze in a very specific way and extract some nugget from, but there is so much more potential there. And I think, actually being able to take advantage of all the data that we generate more efficiently than I know at least I do now, I think is one of the big challenges, probably for many groups, certainly for our group. The good news is that I think we're all thinking about that and we're starting to address it. You see that in the types of people we hire. There's much more. Now the typical student, I think, in the lab is spending half their time at the bench and half their time doing computational work. So that they are certainly changing how they do things. But I still look back and see we generate so much that we really just scrape the surface off of. So I think that's an opportunity. It's not a negative thing, but it's something certainly we need to figure out how to take better advantage of.
The terrific theme in all three of these is about complexity and complexity of biological systems and taking advantage of what we can learn from that. So terrific. Now, you all are renowned scientists, and you come from years, in fact, of focusing on scientific questions. As scientists, what would you like the non-science community to know about plant biology and genomics? What would you say, if you could distill it into the most critical thing for non-scientists to wrap their head around, what would that be?
I could start off with that. I think for me, it was sort of a revelation. I was at a dinner party 2 or 3 years ago, and somebody asked me what I worked on, and I started talking about the plant immune system. And many people just kind of stared at me blankly because they hadn't really thought about the fact that plants have an immune system. And so I guess that's the one thing I'd like to be able to communicate to lay people and to the public, is that plants do have an immune system. It has a lot of really fascinating aspects to it, a lot of parallels actually to the human immune system. And what we learn in plants is, of course, important for making disease-resistant plants, but it also can have an impact on how we understand the human immune system.
I was still thinking. I guess, maybe one of the most important things is to just keep in mind how much potential there is out there. I think it's so easy to go to the supermarket and you see an ear of corn or a floret of broccoli and you think that's it. That is broccoli, that is corn, that's a tomato. And there's so much diversity that we still don't exploit in crop breeding. There's so many opportunities to bring in new traits, to bring in more disease resistance, to bring in different fruit quality, that there's a lot more out there, and that I think also a lot of people, when you think about something different, I know if I always get asked, are those orange tomatoes GMOs, or how about the yellow ones? And all of that is non-GMO. There's no GMO tomato out in the marketplace. So this is all natural genetic diversity that's being brought into modern breeding programs, and there's still just a tiny tip of the iceberg of what's potential that's being exploited.
I think one of the things that has always impacted me a lot is to look back at the underlying commonality that all living things share. And that's the reason why comparative maps work. It's the reason why you can take a gene out of one organism and put it in another. Is because fundamentally living things, honestly, at their base genetic level, they're very similar and they function in very similar ways. And so I wish that the public could understand that we're not so different in many ways. We look and appear different and we emphasize our diversity, but fundamentally underlying that diversity is this incredibly common origin that we share. And that's a very profound thing that genomics has helped us to understand.
Thank you, those were great. Now, switching just a little bit, how are the objectives of plant science research influenced and changing in the context of climate change, in the context of globalization, population growth, and how we are going to feed our global population? So how do you see plant science changing to address these emerging global needs?
It's obviously a really big question, a lot of really huge problems. I guess the one that I think a lot about, because it really does impact what I'm working on, and that is that climate change is going to have a huge impact actually on diseases, because as the climate changes, different pathogens are able to adapt. So we're going to see entirely new diseases start to develop that we just haven't had a problem with in the last 20 or 30 years. And diseases are going to appear in different, further north than they used to, and further south in different crops. So that's going to be a huge problem going forward, and it's due to climate change.
I think we can already see an emphasis on local production, and I think that the return to thinking about being locally a little bit more self-sufficient and less dependent on some grand scale is very important in the face of both population pressure, climate change, and also our appreciation of the varying interests and, let's say, preferences that different consumer communities have. And so it's both out of interest for the diversity, and it's also out of, I think, a need to find a system that has both a local as well as a more global content. That I think that emphasis is a growing one, and one that's quite healthy for us to have a much more direct connection to our food supply, our water supply, and the sources of things that we need to survive.
I think my colleagues have covered these big topics very well. I'll focus a little bit on the fruit. And I think you see certainly the effects of globalization, right? You get summer fruits all year round now because they come from someone else's summer. And they're different, often different varieties. They're growing in different growing regions, maybe different climates, so they're not necessarily the same varieties you would be seeing in the summer here, say if you're looking at peaches or something like that. So that from that perspective, there's certainly a lot more opportunity. There's a lot more things that maybe we didn't have not so long ago. But at the same time, there are, the challenges of growing just, a narrow selection of genetic diversity. I think we probably often appreciate that there isn't the quality you get when you have something locally grown, because you're dealing with all the realities of shipping and transport and storage. It's just hard to get that same quality that you can get from something that's fully matured on a plant that is close by and then picked the day before and brought to you. So I think in the end, what all this represents is just a lot more opportunity for plant scientists like us, because as we understand better the biology of these systems, we'll be able to understand which of these issues we can actually do something about, which are going to be more difficult, which maybe there is no really good solution right now. So I think, these big issues are impacting all of us, and we think about them all the time in our research, and they do certainly change how we focus our research, and certainly where the money comes from to do our research.
Okay, now this is your cue. This is the last question coming from me before we open up the conversation to the audience, and it's intentionally meant to be provocative. And that is, how do you see GMOs fitting into the current research arena in the plant sciences? And tell us if you see alternatives, perhaps, to the strategy for creating plant varieties that we will need to address some of the issues that we've been discussing here.
Well, my research has taken tremendous advantage of GMO technology over the past 25 years. We couldn't have done many of the experiments that we have done without GMO technology. Obviously, commercialization using GMO technology has met a tremendous amount of resistance from the public. And the good news there is that there is a new technology available now. It's being referred to as genome editing. You might be hearing about it on the news and in the newspaper. So it really is a new technology that so far is not being considered GMO. It really isn't a genetic or GMO. It doesn't fit that definition of GMOs. And it allows a very precise change to be made in a plant genome. And so the good news is that it looks like this is not going to have as much regulation and hopefully not as much concern on the part of the public in its use, and it'll be used more widespread.
I was, just kind of jumping to the microphone, because I think in a lot of ways the path that some of the work we do and Greg does is similar. We identify genes that we think are responsible for a process, and we often test that function in a GMO plant. We alter its expression, we overexpress it, and we ask, does it change the trait that we think it might be changing? So as a research tool, it's certainly extremely important, but again, as Greg mentioned, there's been a lot of resistance. Something like tomato, after Flavor Saver, it's been 25 years, there hasn't been another GMO tomato. So there are new technologies, and I think also with more appreciation and more exploitation of available genetic diversity, there's more opportunities for solutions that don't necessarily necessitate a GMO approach. So, and I think practically that's probably where a lot of the seed companies are focusing. If I can solve this problem by using available genetic diversity, why get into the storm of GMO controversy?
I think one of the underlying reasons why GM technology is so considered so essential in some of the big ag applications is because it's easy to patent, because you can see it, because you can prove it, because you can prove it's yours, okay, so you can defend it. So that's very important. What's interesting about the new technology is that as Greg started to say, you can do something very precisely and it leaves no trace, no footprint, no fingerprint. It would look just like nature does it. It's a mutation exactly like what nature does. So I think what's interesting is, will the public, now that the human can do what nature has done before, will the human find that acceptable? Or if the intervention of the human, not the thing itself, but the intervention of the human is the thing the public objects to, will that then be the grounds for further rejection? But what if you can't prove it? So we're entering a whole new terrain, I think, of really complicated concepts about ownership and the defense of patents if this new technology leaves no footprint. And I will say that I think it's both an opportunity and it's very terrifying at the same time. And I think that we should all be quite thoughtful about what it is that we're willing to do. And yet at the same time, I think if you're the one who's faced with the Zika virus, or you're the one who's faced with a malarial epidemic that can no longer be treated by any antibiotic or any, there's no medicine to treat something, would you just sit down and let that microbial population overtake your life? So I just feel like we're at the moment of real ethical turning points, and it's going to take, it's not about whether it's good or bad, it's just going to take a lot of real thought on the part of many, many people to understand when and how and under what conditions we use the technology that the human being is endowed by nature to be able to conceive of and develop, because that's who we are too. Anyway, I find it to be worthy of great thought, and I'm not ready to say it's a brave new world or not. I think we're entering into some very unknown territory.
That was very profound. And at this moment, I'd like to open to the floor further questions that you might have. And I think our panelists might be willing to stay after for a little bit if you would prefer to do one-on-one conversations, for a little bit anyway. But we have our first question here.
Yes, I've been confused about the uproar over GMOs because I've always thought it was a great idea, but it appears that the objection relates more to the kind of chemicals that have been the Monsanto curse, I guess, and that the people are afraid that all of this auxiliary stuff that's going on is polluting the vegetables that we're eating and the crops. And I thought, well, this is a wonderful time to ask this question because I've been very curious. I've always thought the GMO approach was great. So maybe somebody can explain to me why there's such fear and desperation about it.
Yeah, I think you sort of put your finger on it there, is that the first really sort of commercially successful use of the technology was for herbicide resistance. And that did it was associated then with more herbicide use, which was really effective in the field. The farmers, accepted the technology. The sort of downside of that was that there was then a lot more regulations that occurred around the technology, and this partly relates to the public concern about it. So there became more and more regulations, and it actually prevented more widespread use of the technology. It was sort of counterintuitive, but it became so expensive to regulate crops that there were very few companies that could actually do it. So, Monsanto, of course, was one of those companies that were able to deploy the technology, and it sort of stained the use of the technology, I think, in the minds of the public. It just so happened that was the first use of the technology. If it had been used for, proving disease resistance or a number of other things, improving nutrition of crops, and there were subsequent uses of it for those purposes, it might have been completely different.
Actually, let me just add to that. I think the other big use of GM early on was insect resistance in the form of Bt. And the impact globally has been to reduce the use of pesticides by somewhere between 25 and 50%, depending on the crop and the region. So the use of GM technology has actually improved environmental quality and the lives of many, 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 many millions of farmers around the world, many small farmers. In fact, just in case you don't know, the average size of the farm using Bt cotton today is less than a hectare of land. So there's a huge mythology out there that it's only the big farmers that benefit. It's not the truth, but it is in fact the mythology. And now I think it's so confusing in people's minds between the use of glyphosate and then this alternative that it's not one and the same thing. And so if you had to somehow create one monolithic understanding, it's very confusing because it's not one thing or the other. It depends where and how and who. It's like any other form of intervention, right? Too much is not good. But we're at that point where I think it's been very, very, very confusing.
I think part of the problem is that this extends beyond plants. You get this Frankenstein element of, well, we're introducing different species and mixing with humans, and we're modifying DNA and so forth. And part of this goes well beyond the plant development to concerns about what the limits of science are in areas that directly relate to human development and so forth. And some of that fear of the Frankenstein element is coming into the area of plants and vegetables.
I mean, actually, the technology is applied to all forms of life, but so far we don't apply GM to human. But we do apply it to try to improve quality of human life in different arenas, including probably most of the medications you take today. You know, we don't chew on the bark of the willow tree to get our aspirin. We don't, you know, if any of you are diabetic and you have to take insulin, it's GM insulin. So we forget how many of the things that we think are fine are GM technology as well. But I agree with you, that is the confusion, and I think it has implications for, we have an understanding that life was natural before GM, which is another one of those bizarre concepts that we've,
I have actually two questions. It seems to me that the known genetic diversity among all the major crops and the fact that each of them is susceptible to different pathogens that have host specificity, is it possible, this is thinking 10 years from now or 20, to use that information from one main cultivar like wheat to use genes that confer resistance, to put them into corn to confer resistance to pathogens that now can't recognize that because the pathogens of corn have never interacted with wheat genes or whatever, or the products of wheat genes, I mean, in a very diverse way. It seems to me this understanding of all the diversity and then making use of that in a global way could really arrest some of the problems that we have with the ability of microbes to become adaptable to now infect the resistant cultivar 4, 5, 6 years down, as the way we have with rust variants in wheat, for example.
Yeah, in fact, it's not even 10 years from now. That actually has already been done. So there are examples where a gene has been identified in a mustard plant and was transferred to tomato. Tomato, in this case, didn't have any genetic resistance to a certain disease. Of course, you can't cross a mustard plant and a tomato plant, right. So they were able to use GMO technology to move the gene from mustard to tomato, and it gave really effective resistance. There's other examples, too, where...
But it's not been commercialized.
Oh, no, this has not been commercialized yet, but it's being tested. This happened, paper was published a couple of years ago. And there's many other examples in the literature of moving gene from rice to maize to corn and so forth. So the technology is available there again. Probably the main holdup has again been the acceptance or lack of acceptance of GMO technology.
Terrific. My second question is really directed to you. The plants have quite good innate immune mechanisms, but not acquired. And is there any way to introduce into plants an ability to learn from the experience and even transfer that immunity to the seeds so that in the next generation, I mean, that there's some resistance that gets conveyed through the germplasm?
Yeah, that's a great question. We do view plants having an innate immune system just the way that humans do. It doesn't rely on antibodies. But humans also have this sort of system that basically, like plants, every cell needs to be able to respond to a pathogen. So in a sense, that is acquired. They evolve over a longer period of time, but plants don't have antibodies in the sense of they meet an entirely new pathogen and acquire that and it's passed on. Yeah.
Good afternoon. I'm here from San Francisco. I work with some of the big ag growers and processors, and mostly in product development post-harvest. And one of the frustrations I have is everybody in the production business and the marketing business fears getting a label of, oh, this is a GMO product. And so I'm going to go back to Professor Giovannoni's first bullet point on what are the mission, and that is to educate. And particularly when it comes to, I think some of the hysteria about genetic engineering is a lack of understanding. And so I'm going to throw it to you to say, what are you doing to educate people? The EU last week cleared GMO crops that there's no negative health effects from this. And on the record, and yet they still oppose it. They're not allowed to import any GMO products into the EU. So, what standards would you propose to demonstrate safety, both from a plant side, from a biological side? And the bigger issue is other health issues. But is there a standard that would meet your criteria? And how are you going to get that standard out there?
Thank you for the question. I think you hit on a lot of important observations, right? So it certainly is a big problem in terms of perception of GMOs, and I'll just start first with one antidote. So I was at a dinner once in France with the head of an INRA lab. So it's a lab that's kind of the equivalent of a USDA lab. And this was a lab that was doing a lot of GM modification. This was a lab that was promoting the positive benefits of GMOs, very aware of the fact and promoted the fact that there are EU studies, there's National Academy of Science studies, there's many studies addressing kind of the inherent risk. You could probably design something, sure, you could express something that'll cause an allergy, and that would be dangerous. But is there anything inherent about GMO technology that is dangerous? And many have come to the conclusion that that's not the case. So here we are at the dinner, and the wife of the director, who is so positively pro-GMO, says, I will never touch anything GMO because I think it's dangerous. So he can't convince his wife. So some people, I think, are not going to respond to the logical arguments no matter what. Whether it's because, this is food, right? It's something we put in their mouth. That's probably the thing you're going to be most concerned about before everything else. I think in the end, the best arguments, as has been mentioned before, is going to be products, and you're in that area where the consumer appreciates the fact that it's GM, because it's more nutritious, because it has better quality. I think just right now, I understand. I talk to a lot of the seed companies, and they just don't want to touch it. They don't want to be the one to take the arrows. Let somebody else do that. Because there is a lot of negative perception, and I'm not sure that it's as simple as education. There's a lot of people you can explain, and they have other agendas, other reasons why they're not interested in maybe necessarily following the logical arguments. And again, in there, not to be too negative, I think again, because it is so basic, our food is so basic to us, we're probably the most risk averse with that, more than many other things. So that's why a lot of people have a lot of concern.
Is there a standard? Is there a way to come up with a standard to say it's safe?
Yeah.
From a biological standpoint?
I think I can't really address that question, but I think, as Susan alluded to, it'll be really interesting to see how people respond to the editing technology, where it's very, that we use mutation breeding all the time, natural variation all the time. These are going to be changes that will be exactly the same as what you would get through natural variation. Are people going to respond to those as, okay, yeah, that's fine? Are they still going to say, you intervened, so therefore it doesn't meet my standard of what I accept? And it's true, again, you'd never see anything you see practically growing in the supermarket in the wild, right? It's all the product of manipulation. But in the end, the reality still is a lot of people don't want it.
My answer is label it. Label it GM now. We should have done it at the very beginning. We pasteurized milk. People hated it at the beginning. We labeled it. It's fine. But you don't label it. It looks like you're trying to hide something. People don't believe you because you don't tell them the truth. Tell them the truth. It's GM. They'll get over it. They'll try it. I wish we would just label it. I would stop everything else and just put the label on. That's my answer.
We have time for one more question.
So you were speaking earlier about some of the issues with the Zika virus and malaria that's resistant to a lot of antibiotics and other drugs, as sort of a way that something that unifies people behind some of the genetic modification. But I was kind of curious about something such as golden rice, where we see like widespread beneficial impact of this possible variety of rice. And you still see a lot of groups such as Greenpeace like vehemently against these sorts of things. So in the problem of like kind of implementing this technology, are we looking at like a vocal minority that we need to like out lobby or something like that? Is this an issue? I think maybe about universities not being able to go to governments or consumers with information as readily as organizations that aren't governed in the same way that we are. So I'm kind of curious, like, how do you see us changing the minds of not just consumers, who I think are probably a little bit easier to reach, but the people who are really like affecting change policy-wise on these issues?
I want to just say, it sounds like you think golden rice is a product.
No.
There is no golden rice. You know that, right? It doesn't exist as a product on the market. Nobody is eating golden rice anywhere in the world, right? You know that. Okay. So why hasn't it gone out, you mean?
Yeah.
Many reasons, probably. I mean, I know some reasons, but I think my thought is that there's been a lot of attempt on the part of many people on all sides of this argument, on all sides of the fence, to reach policymakers and to discuss in a logical, scientific way. And I actually agree with Jim. I think the fundamental reason that the scientific arguments, the logical, the rational, don't necessarily convince is because the objections are not logical, rational, or scientifically based. So you cannot counteract them with those kinds of arguments. I have an understanding that actually many, I don't know if this is true, but I think many, having talked to many people, I think many people have a deep-seated understanding that comes from Judeo-Christian beliefs, that the world was made in God's image, and any changes are against the law of God. And that's why I think evolution is not taught in some schools. It's the same fundamental clash of some very, very, very basic belief systems. It's not reachable by science or by logic. So I actually believe that that's underlying a lot of the resistance, and it's not something that a policymaker can change. It's not something a university class can change. It's something that people, maybe the reason people change is because they're confronted by a greater evil, and it forces them to reconsider their belief systems sometimes. But as long as you can hold on to your belief system, I know there's a fundamental, deep-seated belief system that probably each of us has. We will defend it to the death. And I think that's actually where, you know, when Copernicus or any of the great discoveries in science, that the Earth revolves around the sun. Things don't revolve around us. Anything that takes us out of the center is somehow very threatening. So I actually believe it's at that level, and that's why it doesn't follow a linear course.
It never disappoints me when I hear conversations among Cornellians. This was a fabulous session this afternoon. I thank each and every one of you for joining us for this thoughtful conversation, and please join me in appreciating our panelists this afternoon.
This has been a production of Cornell University, on the web at cornell.edu.